Welcome and thank you. Uh, Welcome and thank you for joining the 10-year symposium for the Royal-Mach-Gaensslen Prize for Mental Health Research. We are celebrating a decade of excellence in this vital area of research. I'm Chris Carruthers, the chair of the Mach-Gaensslen Foundation of Canada. The foundation honors the lives of Václav Mach and Dr. Hanny Gaensslen by supporting research in mental health, heart disease, stroke, and cancer. The foundation funds many student researchers each year, various research projects, as well as mid-career mental health researchers through the Royal-Mach-Gaensslen Prize. One of the sparks that led to this prize was a talk I heard by former Governor General David Johnston. He pointed out that Canada lacked prizes and awards to recognize our scientists. Often local or national recognition is the first step toward winning international prizes, such as the Nobel Prize. That struck with me as we explored creating a prize for the mental health research with our co-sponsor, the Institute of Mental Health Research at the Royal. From that seed, we now have a decade of growth, featuring some of the top mental health research in Canada. The prize offers recognition as well as $100,000 with no strings attached. Winners whom we surveyed a few years ago highlighted the difficulty in getting funding for innovative, high-risk, high-reward research. The prize is one way for the research to do this kind of research that may or not be funded through granting agencies. Of course, the winner can use the prize money as they wish. Of course, the researchers have already done groundbreaking research to win the prize. One of the most gratifying aspects of the prize is the remarkable breadth of research we have seen. Prize winners work in labs on the front line of service agencies, face-to-face with patients, or in the operating room. They are investing the mental health issues facing children, youth, pregnant people with addictions, first responders, and patients with mental illness. Today, we are happy to have with us the winners, either live or pre-recorded. Over the next 4 hours, you will hear where their researchers have focused their efforts. That might mean a major discovery or getting the research in the community affect lives. I'm sure it will be a fascinating day for fellow researchers and the public alike. To our past winners, on behalf of the foundation, thank you for participating. Your involvement with the prize doesn't end with a trophy and a check. We are hoping that networking among prize winners might spark some interesting ideas and future collaborations. Some of you have mentioned that being a Royal-Mach-Gaensslen Prize winner helped you win other grants or brought attention to your research. We hope winning the prize will continue to open the opportunities for you throughout your career, and we look forward to adding to the ranks of the prize winners for many years to come. Thank you.
Wonderful. Thank you, Dr. Carruthers. That was very insightful opening remarks. So I will now move on to our first ever winner of the Royal-Mach-Gaensslen Prize, and that is Dr. Jean-Martin Beaulieu from the University of Toronto, who will be focusing on intersections between drugs and genetic risk. So I will hand it over to you, Dr. Beaulieu.
And you should be able to see the presentation now.
You're all good. Thank you.
Fantastic. So that's great to be here, even virtually. That was a great honor to get that award. I was very surprised at the time and very happy. And so that's going to be a little bit on the heavy basic science side. So let's go. So essentially, many years ago, I was interested in how some drugs work. And this concept is something that is called, wait a sec, laser, good. So something that is called, so we have kind of two streams of how we cure or find mechanism to cure disease. One is basic research. So we're trying to find the mechanism of the disease, to design a therapy that is addressing this mechanism. And then we run clinical trials, and obviously, hopefully, this will go to the market and help people. For psychiatric drugs, traditionally, the opposite happened, meaning that many mechanisms of action were discovered not in basic research, but efficacy was demonstrated in clinic. And then through reverse translation, people did manage to figure out how some of these drugs worked. And this is how we ended up with generations of antipsychotics and antidepressants, for example. The big missing link in that and very prescribed drug is lithium, for which there is an extensive use still today in bipolar disorder. And many, many years ago, as you can see from what I'm citing, we stumbled on a mechanism of action of lithium that was that lithium, which at the time was known to inhibit a kinase named decogenation synthase kinase 3 or GSK3, was also inhibiting the formation of a protein complex downstream of a dopamine receptor, through which this receptor is also regulating GSK3. And GSK3 and lithium have been shown to act on multiple dimensions, so dopamine signaling, mitochondrial function, inflammation, circadian rhythm, neurotransmission. We still essentially don't know what makes lithium tick or work. And the two questions I had when I started the lab many years ago now is, how generalizable was that mechanism involving this signaling complex composed of PP2A, beta-arrestin 2, and AKT? Keep in mind beta-arrestin 2 for the moment. And what was the mechanism by which itself was affecting behavior, because what we had was changes of behavior in mice. So starting, we try to expand the data to other drug having lithium similar function or therapeutic effect. And we focused on valproic acid, chronic valproic acid administration in mice. And what we saw was an increase in the phosphorylation of GSK3, which inhibits its activity, and the phosphorylation of AKT, which is the outcome of our signaling complex that is regulating GSK3. So we saw that in different brain regions, so that was interesting. And we started to look at, okay, do we also replicate the behavioral signature we saw with lithium? And essentially, we had an effect on locomotion, which at the time people were using to predict anti-manic effect, and different tests predicting antidepressant-like effect. And our surprise is that the highest dose of valproic acid had no effect on the depressive-like phenotype. Both doses had an effect on the manic-like phenotype, but both signature were inhibiting GSK3 equally, which told us either GSK3 is not important, or this may help us find a substrate of GSK3 that is more associated to the behavior than just the inhibition of GSK3. So we went down that way and explored multiple data sets that were available at the time. So one data set was the recently published Schizophrenia Genetic Consortium, GWAS. So we took all the genes that there was 108 SNPs at the time, I think, that were identified. And we compared them toward a known proteomic string of proteins that are regulated by GSK3. And these proteins were also having the particularity of needing to be phosphorylated by something else to be regulated by GSK3, so a conditional regulation by GSK3, which is more or less the conditions we thought we had. And we found one single candidate in all that, which was a RNA binding protein of the family of the Fragile X syndrome protein. So this protein is FXR1 from Fragile X autosomal homolog 1, which is homologous to the Fragile X protein, but which is different in its C-terminal domain and may have different function. And the reason why I thought this was particularly interesting is because of the thing this protein does. So it's an RNA binding protein, meaning it's a protein that will bind messenger RNAs to help to their transport to the different parts of neurons, mostly the dendrites, help to their local mRNA translation. And if you are curious, all things that were published around CAP translation, mTOR pathway, and so on, it's a similar type of function. So this was interesting, and it's even more interesting when one considers a neuron. So this is a picture of a pyramidal neuron, and this little red circle here is the cell body on which everybody is concentrating. And everything you see around is these dendritic arborization, in which most of the action and most of the volume of the cell is. And FXR1 is believed to regulate the translation of protein in the dendrites. So we started exploring whether this was having any importance. So first, is it upregulated in response to chronic treatment? So we did chronic lithium treatment and chronic valproic acid treatment at the 2 doses. And indeed, there is a correlation between the dose that is causing a behavioral effect and the one that is not. The one that is not upregulating FXR1. Lithium and the lower dose of valproic acid are upregulating FXR1. We also made conditional GSK3 knockout mice because the full knockout is lethal, and observed in the cortex when we knocked out GSK3 an increase in FXR1 level. So that was a good indication. We further explored, and I'm cutting a very long story short here, but essentially we found that there was an inhibition of another kinase named ERK at the higher dose of valproic acid, the one that is not causing antidepressant-like effect. And so we searched whether FXR1 could be phosphorylated by GSK3, and the answer was not that much. But if we pre-phosphorylate FXR1 with ERK, the answer was a lot. So essentially, we have a coincidence detection between these two pathways on FXR1. And again, to cut the long story short, this phosphorylation of FXR1 by GSK3 leads to its degradation. So we inhibit GSK3, we increase FXR1. We think that is one of the mechanisms by which the inhibition of GSK3 is causing its behavioral effect. So to dig that further, we developed two types of approaches. So one was somatic knockout using CRISPR technology and viral vectors. And essentially what we do is that we inject these viruses in the cortex or in other brain regions to specifically deactivate the gene we want in the cell we want. So we can achieve very selective neural circuit gene knockout with that in an adult, thus bypassing development, and also use the same virus to overexpress protein. So this is a demonstration of the knockout of GSK3 using this technique. You have here the cell that has not been knocked out. You see GSK3. You have here the cells that have been knocked out. You see a black hole. So we have our detection, and this is an overexpression system to boost the expression of FXR1. So we inhibit GSK3 or we overexpress FXR1. Both would lead to an increase of FXR1. We hit the pathway at two different places. And that gave us behavior that were similar to what we reported before for lithium. So essentially, reduce anxiety and depressive-like behavior in multiple behavioral tests for a similar impact on the emotional disease score. So essentially, a drop of emotional response. By lack of a better word, we're talking of mice. So there's a very, very thin line there to not to overinterpret too much. The fact that we have it in several tests is reassuring. So the question was, do we have something like that in humans? So we went and used a data bank that was available at the time out of Johns Hopkins, which is called BrainCloud. And BrainCloud was an atlas of mRNA expression data in the prefrontal cortex of control humans, correlated, and a collection of genomic SNP. And we went through that, and we did manage to identify two SNP, one strip affecting the expression of GSK3, and one SNP affecting the expression of FXR1. And then we started to look at the interaction between these two SNPs and the impact of that on different human outcome. So this is a functional MRI test with a recording in the amygdala. showing amygdala responsiveness in response to faces that are expressing emotions. And what we saw is that when we have the low expressor of XR1, GSK3 did pretty much nothing. But when we had the high expressor of FXR1, different level of expression of GSK3 were affecting this response to the amygdala. So what does it mean? We also add self-declare data on this control. This is a cohort from the University of Bari in Italy. And essentially, this is a Big Five personality profile test. And in the Big Five, the people that had the high expression allele of FXR1 reported higher emotional stability, unless they also carried the high expression allele of GSK3, in which case this effect was nullified. So GSK3 inhibit FXR1, that's a possibility. Can we expand that to something else? This is a study in the Eastern Quebec cohort from what is now Centre Cerveau in Quebec City. That was a collaboration with the team of Dr. Michel Maziad there. And what they found is an interaction in between the two SNP in the level of symptoms reported in people with bipolar disorder. So, and the interaction explained about 11% of the variance in manic symptom. They were having nothing in the depressive symptom. The cohort was relatively small. We also did another story with the University of Bari in which they test their own cohort and showed again the interaction between the two SNPs, but this time in the context of the severity of negative symptoms in schizophrenia. So again, emotional. And also the severity, the responsiveness of people to on negative symptom to antipsychotic treatments. So there's again an impact. There's an impact in the severity of symptoms when we look at these two SNPs. They also counter-verified that on an additional cohort from the UK Biobank. If you're curious, go see the paper. So our question was then, what is FXR1 doing to neurons? And to do that, we started to look at neuronal excitability in response to FXR1 knockout, FXR1 overexpression, GSK3 knockout, etc. And what we found is that actually the overexpression of FXR1 or the knockout of GSK3 in pyramidal neuron of the prefrontal cortex, we limited ourselves to that, is causing a reduction in the frequency and the amplitude of excitatory potential. Can this explain the behavior? So we decided to modulate the activity of these same neurons using artificial G protein coupled receptor called DREADS. So it's an approach named chemogenetic. And short story, again, these receptors allow to activate or shut down the electrical activity of neurons. And when we shut down or reduce the electrical activity of the same neuron in which we observe these variability of FXR1 and GSK3, that correlate to the behavior we saw before. We replicate the behavioral signature, so reduction of anxiety, emotionality-like behavior. So what is going on there? We wanted to go deeper in the model. So we explore if we challenge the system and do a system like sleep deprivation, what will happen? The reason for sleep deprivation was that issues with sleep is highly comorbid with mental illnesses, schizophrenia, bipolar disorder. Lithium is one drug that has been prescribed for people having problem with sleep disruption. And FXR1 was found in another GWAS for insomnia. So we decided to dig what is happening with sleep deprivation. So mice were injected with an overexpressor of FXR1 or a knockout of GSK3, and they were kept for many days for the genetic modification to take place, and then they were sleep deprived for 7 hours and sacrificed, and the neuron recorded. What we got in the control is a change in the cumulative probability of excitation for the sleep-deprived mice, if we overexpress FXR1, this change is abolished. If we knock out GSK3, this change is abolished as well. So essentially, we have this increased peak amplitude here in the sleep-deprived, and we reverse the effect of sleep deprivation on neuronal activity. We also look at what is happening. So we looked at the cellular or biochemical marker for synapse-express subunits of glutamate receptor, in this case, AMPA receptor. And we showed an increase of AMPA receptor synaptic presence in the sleep deprivation, and a reduction of FXR1 level in the sleep deprivation. So essentially, FXR1 increase, the sleep deprivation increase glutamate receptor synapse expression and reduces the expression of the protein of all FXR1. We looked at FMRP, or FXR FMR1, so the Fragile X protein. The Fragile X protein was not affected. The RNA of another member of the protein, but not the protein of this family, was affected, but in the same direction than FXR1. So that was reassuring that we have some level of specificity for FXR1. And then we looked at the RNA sequencing of what is changing in these neurons in response to sleep deprivation. And long story short, again, if you look at this, these are our control, these are a sleep-deprived animal, and these are a sleep-deprived animal that overexpress FXR1. So essentially, our animals are, the overexpression of FXR1 is reverting the change of gene expression that is induced by sleep deprivation in the context of sleep deprivation. On baseline, that could be entirely different. We did a deeper analysis of the pathways involved and find the top genes involved. And some of these genes, like RAC and VAC and so on, are involved in the trafficking of glutamate receptors. So that was reassuring. And some of these genes were also identified in an interesting type of plasticity model, which is called homeostatic plasticity. And homeoplastic plasticity is essentially a form of neuronal plasticity that compensates for use. So we are familiar with Hebbian plasticity, which is associated with learning. And essentially, the more a synapse is activated, the more the synapse become activatable. But the homeostatic plasticity is the opposite. It's the more a synapse is activated, the more all the synapses in this same neuron will be downregulated. And if a neuron is hypoactive, the neuron becomes hypersensitive. And essentially, you can see that as the reason why learning is not causing epilepsy. We have a reduction of excitability, a global reduction of excitability to compensate. And there are some evidence that there is an association between these mechanisms and what's happening during sleep. And the other reason why it's interesting is because it's an homeostatic mechanism that limits how much a neuronal network can drift in one direction or another. And that brings us to something that people in psychology could be familiar with, which is the concept of allostatic load, which is essentially that if we stress a system, the system will fall out of equilibrium and eventually could go to a maladaptive signal in which it's overloading. And so homeostatic plasticity may be a mechanism that helps neural system to stay in this optimal zone and remain functional. So we used a classic model of homeostatic plasticity, which is to take neurons in culture, prevent their activation with tetrodotoxin or overactivate them with bicuculline, and then look at the surface staining in Western blot and what's happening. So first news, the upscaling, the inhibition of neuronal activity is reducing FXR1 levels. The reduction of neuronal activity is causing an increase in the expression of glutamate receptor at the surface. Bicuculline is reducing the glutamate receptor at the surface. This is the control condition. If we do the FXR1 overexpressor, the treatment with the inhibition of activity by tetrodotoxin is not causing an increase of glutamate cell surface receptor anymore. If you're observant, you will see that our FXR1 in the dendrite is also adopting different structures in the different conditions as opposed to control. So something else is going on there, and we don't know what this something else is yet. In-depth activity of that, the only thing you have to keep in mind really is the end. So what we did is we did a TTX is increasing the peak amplitude of the current of AMPA receptor. FXR1 is preventing this effect. The CRISPR knockout of GSK3 is preventing this effect. The knockout of FXR1 is replicating this effect. And the knockout of FXR1 in the context of the knockout of GSK3 is still indicating that GSK3 is probably upstream of FXR1. And if we force FXR1 to be up, knocking out GSK3 is not going to change much. So if we knock out FXR1, sorry, the knocking out of GSK3 is not going to change much. So essentially what we have is some sort of a system in which there's a pathway that is lithium responsive, that is regulating probably local mRNA translation, glutamate receptor, cell surface expression, global excitability of neural network. And the question we ask for the future is that potentially druggable? So with the help of CQDM and Brain Canada, we ran a small screening for drugs that can potentially affect FXR1 activity. And the answer is yes, we found. So this is an example of a drug that is increasing the translation of a gene. Our reporter gene in this case is, which one was this? Anyway, essentially, we are able to affect the regulation of mRNA translation by FXR1 using small molecule drugs. Whether this is potentially usable, we don't know. Right now, we are at the stage of trying to get good tool compounds and see what's going on. So I would like to thank people that were involved in this story. From my lab, Thomas, who did the initial work, to Jivan, who did the work on the FXR1 and homeostatic plasticity. Alicia was our electrophysiologist. Deepa is continuing the project now, and different other people involved. Collaborator in the TOT lab, now at University of Ottawa, with whom we worked with the electrophysiological characterization at the beginning. The Montgrain lab at University of Montreal, which helped us with sleep deprivation and which performed EEG analysis in the same mouse, which I didn't present today. Collaborator at University of Barrie, collaborator in Quebec City, and ongoing collaboration with the Liber Center for Neurodevelopment at Johns Hopkins, with Gianluca and Danny Weinberger. So, and our ever on again, off again love affair with our different sponsors. Thank you.
Wonderful. Thank you so much, Dr. Beaulieu. It's fascinating work and sounds like you're on a great path. So I will now introduce our second Royal-Mach-Gaensslen Prize winner who received the prize in 2016, Dr. Rudolf Uher. Let me just swap this here, who will be discussing depression prevention and personalized treatment. So over to you.
Well, hello, everyone. It's absolute pleasure to be here. So amazing to see so many people very interested in mental health. I'll just share my slides. Hope that's working. And it's my pleasure to speak here about depression, which is my main and favorite topic in research. And I'll specifically talk about prevention and personalized treatment for depression. In terms of disclosures, I don't have much to say except editorial fees from JAMA. All the research that I'm going to show has been funded by either independent philanthropy or competitive research funding sources, and that includes the RMG prize. Depression comes by many names in the current literature. The major depressive disorder is the preferred term, but for much of history, this has been a part of slightly broader concepts like manic depressive illness or melancholia. What people with depression are experiencing sometimes sounds different from the terms that our textbooks use to describe it. People with depression talk about feeling disconnected, heavy, stuck with no control. So that's often the language that you hear, and the essence is that this stuckness lasts for weeks, months, sometimes years. And a somewhat unfortunate thing about depression is that everyone seems to have an opinion. So people living with this difficult condition often get a lot of unhelpful advice. And the biological tropes and simple explanations are just as bad as the alternative and natural ones. It's one of the most common kind of issues that people with depression report, that they just afraid of mentioning anything because they keep hearing these things that do not help make them feel better. Well, what we know about depression is that it is brain disease, and the answer doesn't come from brain studies the way you imagine it. It comes from genetic studies, the most conclusive answer. And I have to show this. It's one of the largest experiments in medicine done ever, and it's just been published a couple of weeks ago, earlier this month, in Cell, the largest ever genetic study of any mental disorder. Includes 5 million human participants with genome-wide genotyping. And this powerful study has shown us more about depression than anything we have known before. Over 300 genes linked to depression in a very conclusive ways, and which genes and where they are and what they do, it really tells us so much about depression. And the bottom line is there are brain genes. There are genes that are involved in formation of synapses with the connections between neurons, release of neurotransmitters, the communication molecules between brain cells. And also, actually, it was interesting to see molecules like estrogen receptor figuring high among the most strongly associated genes with depression, knowing how depression is to a degree sex-specific, more common in females and often tied to reproductive stage. This study told us much more than that. We call this Manhattan plot because of the many skyscrapers. Each of them shows a part of the genome that has something to do with depression. Anything that's above the red line is like well beyond chance association. So you can see there are many, over 600. And you can use the bulk of these depression-associated genes to see where in the body they work and they are expressed. So on the bottom center, you see something that looks like it comes from like a brain scanner. Well, it doesn't. This is of the whole human body, the parts of the human body where the depression-related genes are most expressed. And it's not just that it's in the brain and in the neurons, but it actually points to the part of the brain called anterior cingulate that we know when we stimulate it, we can cure some of the most treatment-resistant cases of depression. So you get this from genetic scan. Another amazing thing, we were able to see all potential medications and drugs for which one may be interacting with the genes involved in depression causation. And it's incredible. The top comes, unsurprisingly, antidepressants and antipsychotics, but also medications like pregabalin that are used for pain and for anxiety. perhaps most practically significant, these large genome-wide scans enable creating scores that help predict depression risk at the individual level. And now for the first time, we're able to do it in people of all ancestries from all continents in Earth. And that this is a major advance, knowing how different the structure of genome may be. So we know a lot about depression now that we did not know even just a few months ago. But the real reason I like to work with depression, I want to work with depression clinically and research-wise, is the massive impact depression has on humans, on people living with depression, on their families, close ones, and on the society. The rose-like figure here shows you that depression is the most common reason for disability in every single age group starting from age 15 onwards. So this big chunk of disability of all types of mental illness here, it's due to depression. Depression is actually the second most common reason why people are not able to work, second to backache only. And that's like of all medical diseases. And depression has very serious impacts in terms of individual. When someone develops depression before the age of 19, they are twice or more likely to develop a long list of serious medical illnesses, including diabetes, liver, kidney disease, cerebrovascular, heart and brain vessel disease, and also serious infections and having accidents and things like that. They are 6 times more likely to die young. So depression is a deadly disease. And the question is, can we change this? I believe we can change this best if we look at depression from a life course perspective. When in the human life it happens. Here, the first thing you see is that the early ages are extended and the later ages are compressed because so much happens in the early stages of life. The red line here is my attempt to draw the development of human capacity. Think intellectual, think social, all that humans can do. It's this massive development in early childhood, and there's another steep period of development in adolescence. And I believe it's no coincidence that it is just about the time when humans are just about to reach their top potential at the end of their teen years. That's the time in life that they are also most likely to develop depression for the first time. Well, this onset of depression, as we call it, is usually not the first manifestation of any mental health problems. There's often early problems like anxiety that often happen in childhood or early adolescence, or irritability or defined behavior in even younger children. And they all precede and predict more higher risk of depression. So we call them antecedents. And once depression develops, it also can follow multiple paths. Many people who experience first episode of depression, this will remain their first and only experience of depression for the rest of their life. But others experience many recurrences or relapses, or even persistent depression that not really remains in episodes, it just continues on and on. Depression is also often the first manifestation of what will eventually become bipolar disorders or psychosis, so potentially even more serious types of illnesses. And then it can lead to the massive physical morbidity or premature death. And it is in this schema that it's good to look for opportunities about how we can change and diminish the impact of depression. Well, I believe the first opportunity is here. It's among people who perhaps have had some of the early manifestations of risk in childhood. Can we stop them or some of them from developing depression in the first place? Can we preempt depression? And this is about prevention. And a number of researchers have tried. And what we know from summary of these findings is that prevention that's targeted to individuals with identified risk, it's much more successful than anything we can try to do universally. The kind of mental health in schools has very iffy problematic impacts that may be positive in the short term, but seems to completely disappear within a year or so. Here, long term refers to anything lasting a year or longer, and you can see that targeted prevention maintains its effect in reducing depressive experiences, whereas the effect of universal preventive efforts seems to have disappeared within this time. So we know that targeted prevention can make at least some difference that lasts. Here is an example of possibly the best study in the field, about as good as it gets. Here, Dr. Garber and colleagues selected young women, adolescent females, aged 13 to 17, who were daughters of mothers living with depression and also had significant subthreshold but depressive symptoms themselves. And some of them were offered cognitive behavioral prevention program, and others remained in what's being called usual care, but it's really nothing, because there's usually no services for things that don't reach the threshold for diagnosis. And over about a year, many of those in usual care, actually 1 in 3, developed major depressive episode. And the rate of developing depression was reduced to about 1 in 5 in those who received the cognitive behavioral prevention program. It's a significant reduction. Like you could say, many young women did not develop depression because they received a preventive program, but it is still shocking how many of them did. And it raised some question about the usual care possibly not being the best thing, especially after you are identified as being at risk and included in a project like this. We looked in detail into the early identification of risk, because not everyone who's going to develop depression has a mother living with depression. So we synthesized a lot of experiments. We synthesized almost 600 relationships between potential antecedents and outcomes, and we found 9 early predictors, antecedents, that predict depression onset over years to come, and independent of context and family history. And there are things like anxiety and affective lability, but also unusual experiences and early sleep problems. And when you take them together, depression is very much predictable. I know it often feels like coming out of the blue sky from those near the person, but when you look across large groups and you ask enough questions, there is a great degree of predictability. We are in the process of testing an intervention focused on these antecedents, and being tested in a way that does not require the allocation to usual cares of some people who are told they are being at risk, so really avoiding the potential to harm someone. And with some slowdown in the year 2020, 2021, I'm glad that the numbers receiving the intervention have recovered, and we nearing 200 young people who have received these interventions in Nova Scotia, and they are all being followed up so that we can determine the long-term effects of these prevention efforts. Now back to the lifetime view, we focused on the relationship between antecedents and the onset of depression when we talked about prevention. What about changing the relationship between the initial presentation of depression and all the various outcomes? Can we shift more people towards the sustained recovery path and away from the more serious and adverse outcomes? And the way we're trying to do it is through personalized treatment, following the simple question, what works for whom, or which treatment will work for which patient or which person with depression. A lot of this work is happening within CanBind, a Canadian network of clinicians and researchers interested in biomarkers and treatment for depression. And as part of CanBind, we have made a couple of pretty significant discoveries, I believe, predictors of who will benefit from which treatment. Here, the blue line shows how people with low and high levels of specific symptom profile respond to a typically used antidepressant called escitalopram. And people who score low on this interest activity symptoms improve by a lot, and people who score high improve by a little. And one idea was, well, people who don't improve by a lot, do they need something else or additional? And when we added a second medication called aripiprazole, also known as Abilify, we actually added a little bit of benefit for everyone, but almost twice as much benefit, or yeah, twice as much benefit for those who were at the higher level of these symptoms. So these are people who often don't feel the interest, the motivation, and who remain inactive for a lot of time when they are depressed. This is during their depressive episode. They really seem to need this add-on medication because we pretty much know that they will not do well with escitalopram alone, with an antidepressant alone. We found a second predictor, and that's work led by my colleague Farnak Farzan and Ben Schwartzman at UBC, that again shows much less response to an antidepressant and much more relative benefit with add-on among people with specific brain activity profile measured with EEG, electroencephalography. And you can see that these two pictures, they look kind of similar pattern in terms of how they predict response to treatment, but they actually not measuring the same thing. So when we bring the two together into a predictive algorithm, we get an even stronger prediction of who will need this add-on treatment to get out of their depression. And here the effect became so strong and so robust that when we talked with people with depression, they say, why wait? If the standard is you always start with an antidepressant and then you only do add-ons if the antidepressant doesn't work well enough. So why should we be put through weeks of failure? Let's test it straight away. What about giving people the combination treatment from the get-go? And this is actually a personalized treatment that is currently being tested in a large Canada-wide trial, Optimum D, recruiting currently patients across 9 sites in Canada. And the experience with the first 80 patients who have been through this over the last year suggests that the outcomes are improved remarkably compared to standard treatments for depression today. We would like to take this further. If we can personalize and give people something that works for them before trying and without having to go through things that don't work for them. What about doing it for people who experience their first episode depression? I'm really excited about this because I feel this has even much more potential to change the life trajectories of people with depression. If a young person, perhaps someone in their late teens, becomes depressed, the difference between getting a first treatment that works within a few weeks versus having to try a few and taking months to recover. That may be the difference of graduating with their cohorts or not, or getting to university or not. And important may be the difference between their depression remaining a single episode in my teens versus becoming a fluctuating, recurring chronic illness. So we call this TIDE, Treatment Interrupts Depression Early. We started it in Nova Scotia. Very happy that we just in the process of opening a second site in Quebec with generous support from Behavioral Health Foundation, Medavi Health Foundation, and Hewitt Foundation. So fully philanthropically supported project. And young people between age 12 and 24 experiencing their first episode depression, receiving the best possible treatments with antidepressants or psychological treatment, whilst they're providing a lot of information. Again, we're using electroencephalography to measure brain activity. We record brief speech that in even 1 minute gives so much information as we could previously get from a 30-minute long interview. And actigraphy to track activity and sleep in real time. And with all this information, we hope to get similarly predictive algorithms for who needs which treatment will lead to faster or more sustained recovery in more young people. So with this, I would like to conclude what we have discovered and know for sure is we can predict depression, which means targeted prevention is possible. We know we can predict treatment outcomes, which means personalized treatment is possible. And with the combination of prevention, targeted prevention, and personalized treatment, I believe these are the tools to cut the burden and costs of depression for the next generation. So I'll stop here. I would like to thank the many people who have been involved in these projects and the funders who have made them possible. And thank you so much for listening and being here, everyone.
Thank you so much, Dr. Uher. It's fascinating work that you're doing, and you're on a great path forward. So now we will pass it along to our 2017 recipient, Dr. Benjamin Goldstein. So I'll just ask that you turn your camera on, Dr. Goldstein. Excellent. So we can see your slides, and you are good to go. Thanks.
So thank you so much for this opportunity. What a great opportunity to hear from everybody else on this panel, and just a comment about how inspiring the existence of this award is for the entire field in Canada. I have no commercial interest to disclose, and what I'm going to be focusing on is the connection between bipolar disorder and cardiovascular health, and whether it's time for clinical action, which I think is a self-evident declaration, but what I hope to do over the next 15 minutes is to convey to you why I think that's the case. So to start with, I'm presenting epidemiologic data. So these are representative population sample in the United States of over 40,000 adults. And what you can see is in this two-wave study that was separated by 3 years, what was the age of adults who had new onset cardiovascular disease? So this is the individual reporting that a doctor diagnosed them with heart disease. And as you can see, that the age of adults with bipolar disorder type 1 was fully 17 years younger than controls. And it's important to note that in this study, controls could have anything except for bipolar disorder or major depression. They could have anxiety disorders and addictions. So the question arises, if this divergence from what's expected in terms of the age people have heart disease is already evident 17 years early, when did the biology that underlies this process begin to go off the rails? And another fact that you see in this slide is that while there's some degree of a diagnosis-related pattern, meaning the prematurity is greater in bipolar disorder, you see the same thing in major depression. And I emphasize that this is a population study, and you saw from Dr. Uher's slides how the genetics of depression also relate to heart health, you see the same thing with other serious and persistent mental illness. It's important to note that in the previous slide, the association between heart disease and bipolar disorder was over and above what could be explained by blood pressure, smoking, obesity, and self-reported nutrition and physical activity. So it raises the question of what else is driving this, if not those traditional cardiovascular risk factors. So here I highlight the fact that Emil Kraepelin, who was, one of the leading theorists about bipolar disorder, and he really benefited from having a keen eye observationally and not having a conceptual framework, but rather just documenting what he saw. And indeed, if you look back to his seminal text from over 100 years ago, he noticed this. So he says, not at all infrequently, and in comparative youth, arterial sclerosis is present. So this is not just a function of contemporary medications or lifestyle or the obesity epidemic, but rather something that appears to be long associated with bipolar disorder. For those who may not be familiar, Aaron MacKillop at the University of British Columbia runs an Ask Me Anything event on Reddit every year on World Bipolar Day. And so various experts are asked to address questions that are posed in their subject area of expertise. And so this question came from an individual, and I'll read the full quote. So, hi there, I'm 52 with bipolar disorder. I don't drink, smoke, do drugs. I was in good shape. There's no history of heart disease in my family, but then the preceding April had a massive heart attack leading to heart failure. It turns out this individual was told that he had started developing severe coronary artery disease at some point in the previous decade. And that's what he was told. As I'll convey to you, I think probably it was before that. I've read up on the correlation between bipolar and heart disease, and I assumed that lifestyle factors were the driving force, but it doesn't seem to be the case in my situation. So the question is, what's the current thinking around bipolar disorder and heart disease? So I think that this case exemplifies why it's important to consider the direct association between bipolar and heart disease, because if it's just stemming from smoking or obesity or lack of exercise, I don't think that's as surprising. Whereas if there's a direct association, it really, provokes us to think critically about, should there be different types of treatments? Should we be conceptualizing bipolar disorder in a different way? One of the most impactful outputs from our group's work, I think, preceded the receipt of this award, and that was publishing a scientific statement acknowledged by the American Heart Association in one of the leading international cardiology journals called Circulation. And what this paper did was reconcile a disparity that existed in the literature. There had previously been another scientific statement that positioned various medical conditions that affect young people that drive excess rates of heart disease later in life and accelerated atherosclerosis. And on the basis of the benchmarks that they had set, it appeared that major depression and bipolar disorder in youth should have been included. And this paper in 2015 allowed us to reconcile that disparity and add to the list of conditions that drive early and excessive heart disease, these mood disorders. And I think what's important, in addition from a psychiatric perspective, but from a lifespan perspective and from a preventive cardiology perspective, is that mood disorders in young people affect 1 in 10 of them by the time they reach the age of 20. And so really this is a issue of substantial magnitude at a population level. So with that brief overview of the epidemiology and the implications as it relates to specific early intervention and prevention approaches in young people with mood disorders, the question is, what does this have to do with mental health? And so what this slide does is summarizes a number of studies, and all of them point in the same direction, which is if you have one or more cardiovascular risk factors, be it hypertension, high blood pressure, diabetes, obesity, and especially the confluence of multiple of those, invariably, almost all of the studies that have examined this have found that the more cardiovascular risk you have, or the presence of a cardiovascular risk factor, the worse your psychiatric illness manifestations are, in this case, bipolar disorder. So in people with bipolar who have one or more traditional cardiovascular risk factors, they have more mood episodes, they have more hospitalizations, their functioning is worse, more addictions, more suicide attempts, and I've put asterisks beside the findings that have been replicated in both clinical samples and epidemiologic community-based samples, also both in youth and in adults. So that suicide attempt association with cardiovascular risk is highly replicated. And while we can't say anything about causality, it certainly behooves us to focus a little bit more about what may be driving that link. And so that's the question is, right now, most healthcare settings, and I think this is true internationally, don't intentionally target heart health for the benefit of mental health. And the question is, could that help our patients in a context where we have relatively few novel treatments coming down the pipeline? So with that, what I'm going to do is turn to a series of snapshots that our group has undertaken studies with the intention of showing the relevance of this association early in life. Obviously, I am a child and adolescent psychiatrist. My average age of my patients is 17 years old. They don't have heart disease, but they are at risk for future heart disease. And so these series of studies show how we can demonstrate the relevance of heart health even at this early age. So in this first slide, what you see is the association of bad cholesterol, quote unquote, LDL, with hippocampal volume. And if you alert your focus to the orange line and dots, what you see is that with increased LDL levels, there's smaller hippocampal volumes in teens with bipolar disorder. There's a trending significant association the other way for healthy controls. What I'll say is that if this was a middle-aged or elderly sample, this would be no surprise. This would be something that's been shown dozens, if not hundreds of times, that more cardiovascular risk factors, in this case lipids, are associated with smaller brain structures in adults. This just hadn't been done in youth before. And what you see here is that even at the average age of 17, in teens with bipolar disorder, but not their healthy comparison group, there is this association that's already manifesting. So higher LDL, smaller brain structure. So you can make the argument that although LDL is largely genetically driven, there's also behavioral factors, nutrition, etc. So another way to approach this association is to look at genetics, which Dr. Uher focused on. And we looked at polygenic risk scores. So these are global genetic indices of one's propensity for a given condition or a given trait. And we focused on coronary artery disease and also myocardial infarction. So what you'll see here is the healthy controls in blue, the teens with bipolar disorder in red, and I'm sorry, the blue is the overall sample, the red is the teens with bipolar disorder, and the green is within the healthy control group. While these regions are in different places around the brain, and while the significant findings that we detected were related to different imaging metrics, so there's volume, area, thickness, the main point that I would make is that you can see here the consistency of the association, regardless of the group and regardless of the region and regardless of the neuroimaging metric, which is to say that the higher your molecular genetic risk for coronary artery disease, the smaller the structure of regional of brain structure regions. And in addition, you see that the number of regions that survived the stringent correction for multiple comparisons is more common in teens with bipolar disorder, where we detected 5 regions. Now, that's not a statistical comparison, but I think it just is worth commenting on. The other thing that I'd comment on from this data is that these findings are more pronounced in females with bipolar disorder. And that's important because we know that the extent to which bipolar disorder drives excess and early heart disease impacts women more. And while there's a lot of gender-related factors, this suggests also that there may be some sex-related factors that are, upstream primary biological effects, and highlight for us that in addition to reconciling disparities in access to treatment and preventive interventions, we really need to be thinking about this group of young women with bipolar disorder as a particularly high risk for the cardiovascular-related consequences of the illness beginning in early life. So moving along, the next approach that we take in our research focuses on imaging of the eyes, of the retina, the back of the eye. So what you can see here in this nice picture on the right is a picture of the retina. And as you can see, it's well perfused with vessels. And this is analogous to the brain. So as you see in the retina, there's a lot of blood vessels and a nerve. In the brain, there's also a high density of blood vessels. And while we can use MRI to glean proxy indices of cerebral vessels, this is a way to directly visualize central nervous system blood vessels. As you can notice, most likely on your screen, there's wider, darker vessels and narrower, lighter vessels. And the wider, darker ones are tiny veins or venules, and the narrower, lighter ones are arterioles or tiny arteries. And there's been a lot of studies, especially in adults, showing that if you look at the caliber or the width of these vessels, especially the ratio one to the other, so typically the greater the ratio of your arteriolar diameter to your venular diameter, that's associated with more healthy indices from a cardiovascular and a brain perspective. It's been much less studied in youth, and it's been much less studied in psychiatric populations. But we were interested in this as a proxy that we could use to estimate cerebrovascular health. As you can imagine, going to an MRI scanner, which we do for a lot of our research, it takes a lot of time, it's a lot of money, it's not necessarily accessible. whereas the apparatus on the left, as you see here, this work was led by Dr. Megan Mio, who's now a postdoctoral fellow with our group. This can be done in under 10 minutes, ambulatory, just walk in and get it done with no drops. So what you can see in this slide is we looked at the association of retinal vascular caliber with cerebral blood flow. So generally speaking, it's well acknowledged that the brain has high metabolic demands and it has a high need for oxygen and blood. And what you see here is that the retinal vascular index thought to be good, so higher arteriolar to venular ratio was associated with higher regional cerebral blood flow. And this was, as you can see from this slide, diffuse around the brain. So what does that tell us? There's still value, obviously, to examining cerebral blood flow for a number of different hypotheses to test. But a simple photograph of the retina does give you an index, at least within the bipolar disorder group, of the extent to which the brain is being perfused. So finally, I'm going to give an overview of our cardiac MRI research. And what we did was we modified an approach that had been taken by Matthias Friedrich, who's a renowned imaging cardiologist at McGill. And what this task involves is, just like you would take a young person to an MRI scanner and use fMRI or functional MRI, either during a task or at rest, you can do the same thing in the heart. And you get same types of contrast. So if you look at the fMRI bold signal, it's oxygen sensitive and it responds to changes in blood flow. And so we looked at the bold signal in the heart in teens with bipolar disorder and healthy control teens. The task was, as you can see from this curve, the first dot farthest on the left is an acquisition at rest. The second dot at 10 seconds is just after they had completed a hyperventilation task. So they had 1 minute where we had a paced breathing regimen. So deep breath in, deep breath out repeatedly for a minute, and then you exhale, expunge all of your carbon dioxide, and expel all of your oxygen, and then so begins the accumulation of carbon dioxide, which is a potent vasodilator in the heart and in the brain. And what you see reflected in the curves is a time-related change in the intensity of the MRI signal. So this is a proxy or an index for increased blood flow. And you can see that in both groups, there were significant increases. So as you're holding your breath up to 40 seconds, there's an increase in the intensity of the MRI signal in the heart. But what you'll also see is that the curve is more blunt or flat in the teens with bipolar disorder. And this was significant as of the 30-second mark. So the way we position this as a proxy for microvascular function in the heart. I don't present the data today, but we also looked at more traditional indices like the volume of the left ventricle or the ejection fraction. So these are the types of indices you'd get if you're going to get evaluated for heart failure. And there was no differences between the teens with bipolar disorder and the healthy teens in those traditional indices. Whereas this hyperventilation and then breath hold induced microvascular metric does distinguish the two groups. And the magnitude of this effect is about halfway between healthy individuals and what you see in people being treated for heart disease. Because it's a proxy index, it's fMRI, it's relatively novel. These are teens that don't have heart disease. We wanted to make sure that we bolster it in biology. And so what we did was looked at whether it maps onto genetic risk, molecular genetic risk for heart disease. So as you can see here, that same index, the extent of cardiac microvascular dilation, the greater your genetic risk for heart disease, whether it's myocardial infarction or coronary artery disease, the less robust your coronary microvascular reactivity is. So we think that even though we're not doing a direct visualization of atherosclerosis, this is really quite a promising proxy for early indices of heart disease risk in this population. So with that, it completes the round of different snapshots. So looked at the brain, traditional cardiovascular risk factors, the eye, the heart. And all of this to say is, as Rudolf was mentioning earlier, while bipolar disorder is defined by brain-related symptoms, behavior, emotion, cognition, it really does appear to be a systemic illness. Our position is that to some extent, this is a cardiovascular illness, and especially a microvascular illness. And the next question, I think that the work so far has provided some support for that assertion. The next step is to say, who are the individuals, who are the youth that have a convergence of cardiovascular risk factors and for whom specifically targeting cardiovascular risk is really an imperative while they're still young. And so that's something in terms of looking at people that are especially predisposed that we're interested in. So conclusions are that youth with bipolar disorder have increased cardiovascular risk from a convergence of factors. You heard of traditional cardiovascular risk factors, but also microvessels and how they impact the body. Cardiovascular metrics are associated with psychiatric symptoms and brain structure and function in teens with bipolar disorder, already at an average age of 17. There's potential value for optimizing heart health in teens with bipolar disorder, not just for their emotional health, but for their long-term brain health and its retention and for heart health. And my view is that the heart bipolar link can, in addition to guiding preventive cardiology, help identify causes of bipolar disorder, give us the motivation and the impetus to try new treatment approaches, and also very importantly, to reduce stigma. And I think that although there's been improvements in stigma, the separation of mental health and physical health, so to speak, has perpetuated to some extent remaining stigma. And just a note to say that the work that I have presented today is focused on bipolar disorder. We got the great news today that we're going to be supported by CIHR for the next 5 years to extend this to serious mental illness, so major depression, bipolar, and psychosis. And this will be a really exciting collaboration with McGill, Sick Kids, and colleagues at Sunnybrook. So I want to acknowledge our funding organizations and especially our youth and their parents who, permit them to do these intensive studies, and the fabulous team at the Center for Youth Bipolar Disorder. Thank you for your attention.
Thank you so much, Dr. Goldstein. That was fascinating, and congratulations on your new funding. That's incredible news.
Thank you.
Okay, so we will now move on to our 2018 recipient, Dr. Tracie Afifi from the University of Manitoba, who will focus on child abuse, mental health, and substance use in Canada with a focus on sexual identity. So over to you, Dr. Afifi. Oh, you're just on mute.
Okay, how's that?
Great.
Just want to confirm that you can see my slides.
Yes.
Okay, great. All right, thanks for having me. It was thrilling to get the invitation to bring all of the previous awardees together. It's a great idea, and it's been lovely to learn more about everyone's research. So, wonderful idea. So I'm going to be talking about child maltreatment, mental health, and substance use with a focus on sexual identity and a focus on younger individuals. So really that emerging adulthood phase. So just want to acknowledge that I'm at the University of Manitoba, so I'm on Treaty 1 territory. And I also just wanted to mention that for my talk, I will be talking about things like trauma and abuse, neglect, and serious injuries, and just want to acknowledge that this can be difficult topics to talk about and to hear about. So if anyone needs to leave the presentation at any time, feel free to do that, or come back at any time as well. So just wanted to make sure that people know the nature of this talk. I'd like to make some acknowledgements for the people that work specifically on this project. So the list of my co-authors are there, and also others who were involved in the funding of this work. And I will be presenting one specific presentation today, but it's sort of part of a big group of studies that we've been doing, and sort of has informed new research that we're currently looking at now. So this particular study that I'll talk about for the next 15 minutes or so is currently in press at the Canadian Journal of Public Health. So the objectives of the work that I wanted to focus on today was really looking at trying to better understand the prevalence of child maltreatment, mental health, and substance use among emerging adults. So the age group we were looking at was between ages of 18 and 23. And we wanted to focus on diverse sexual identities. So why is that important? Well, what we know from past research is that there's a lot of sex differences in the child maltreatment literature, and also related to mental and physical health outcomes. So we know there's a lot of differences based on males and females, But what we don't really understand to date is really looking at how child maltreatment is related to sexual identity, and then how those experiences are also related to depression, anxiety, and things like at-risk alcohol and cannabis use. We also wanted to make sure that we were looking at emerging adults, because this is a time when substance use may increase, and where depression and anxiety may start to become problematic for individuals. And it's also such an important pivotal time in the developmental lifespan. So for the study that I'm presenting today, we wanted to compute the prevalence of sexual identity using a community sample. Which is important because a lot of the work in this area, any work that's been done so far, sometimes uses samples recruited from social media or university samples. So we wanted to kind of get a better quality sample. And we also, as mentioned, wanted to focus on emerging adults. So often this research is looking at adults and who are older in age and not sort of right at that point where they're moving from adolescence into adulthood. We want to determine the associations between sexual identity and child maltreatment. We wanted to look at many child maltreatment types, because often this research is limited to sexual abuse. And often when we look at child maltreatment in Canada, we only look at one or two types of abuse. And so with this data, we're able to look at many different types of child maltreatment experiences. So it also advanced our knowledge by looking at that. We wanted to look at the associations between depression, anxiety, alcohol, and cannabis use, and we used validated tools for all of those things, as well as for measuring child maltreatment. So that's also something that's often not done in this literature. And then we wanted to look at moderation effects or interactions between these experiences of child maltreatment and sexual identity on these mental health and substance use outcomes. So really wanted to look at whether or not things are worse when child maltreatment occurs, and also thinking of whether or not things are worse for those who have who identify as a sexual minority. So the data we used for this study is called the Wellbeing and Experience Study, or we call it the WE study. So this is data that's collected, that was collected from my lab. We started this work in 2015, the idea of it, and got funding for it around that time, and started to develop this as a longitudinal intergenerational cohort study design. And so since that time, we were able to collect 7 waves of data. So among 1,000 Manitoban youth at baseline, and that was paired to a parent. Usually it was the mom, but it didn't have to be the mom. So a person most knowledgeable, which is, for those who know StatsCan language, the person most knowledgeable of the child was welcome to be the parent to answer the questions related to relationships and experiences. So at wave 1, the youth were 12 to 17 years old, and we started data collection in 2017. And then we proceeded to collect 5 waves of data collection of youth approximately annually after our baseline data were collected. We ended up collecting 2 waves of data for the parents. So we collected it at baseline, and then we collected it again in 2022. And so it's a longitudinal sample, it's voluntary, and it relied on self-report surveys. And we both had urban and rural families in Manitoba. It's not a representative sample for Manitoba, but it did map onto sociodemographic factors at baseline. So it's a good representation from a community sample from across Manitoba. So the variables included in this study were sexual identity, child maltreatment, as I indicated, measured with valid tools, and looked at a number of different child maltreatment types, which we'll talk about. We had sociodemographic covariates, and then the mental health and substance use we focused on was depression, anxiety, at-risk alcohol use, and at-risk cannabis use. So we used descriptive statistics, and then also used logistic regression analysis to look at the relationships between sexual identity and each child maltreatment type, but also looking at the outcomes of depression, anxiety, at-risk alcohol, and at-risk cannabis use. We used interaction terms to understand interaction effects, or whether or not these experiences worsened the relationships. And then we also did some ad hoc analysis as well, because we only have a small sample here of 1,000 youth at baseline. And we used data from across our waves. So we had less than 1,000 when looking at multiple waves of data. So, and we're also looking at child maltreatment experiences in a community sample and sexual identity. So we did have limited power with this study. So at some points we did have to collapse things and do ad hoc analysis just to understand if the trends seemed to be supportive of what we see when we thought our models were perhaps underpowered. So in terms of our results, we have, this is just our descriptive table. So you can see some of our sociodemographic characteristics in our total sample, and then also by our sexual identity. So here we just have it dichotomized as heterosexual, straight versus other identity. And in our models, we were able to look at it in more detail, but just for descriptive purposes, you can see what our community sample looked like. And these are the age at wave 5, so ranged from 18 to 23 years old. So the first set of data, what I'll show is I'll orient you to the table. So I looked at the associations between sexual identities. So you can see them listed down the side of the slide there. So homosexual, gay or lesbian, bisexual, other identity, and those indicating I don't know. This was in comparison to our heterosexual or straight group. So that is the line, the gray line there that you see along the number 1. And then we've graphed our odds ratios for whether or not the relationship between sexual identity and physical abuse among our samples. So this is all for physical abuse. And you can see that we only had one significant relationship here, but it was with bisexual sexual identity, increased the likelihood by 2.39 times increased odds that they would also experience physical abuse. This slide is the same, but it's changed for sexual abuse. So we can see that the significant relationship was also found for the bisexual sexual identity and sexual abuse by an increased odds of 3.66 times. And this slide is for emotional abuse. Again, we're seeing only significant findings for our bisexual sexual identity category for increased likelihood of experiencing emotional abuse. And here we have, for emotional neglect, we have 2 significant findings. So that is for bisexual identity and other identity. So these 2 identities compared to heterosexual increase the likelihood of experiencing physical neglect by 3.6 times for bisexual identity and 3.36 times for other identity. But what you can note here is that for homosexual and I don't know, while it didn't reach statistical significance, we have odds ratios of 2 for I don't know, and odds ratios for 3 for homosexual. And you can see that line is quite long. It does cross 1, which means it's not statistically significant. So here, and with some of our other findings, we do think that this is probably just underpowered models, and that these other sexual identities are probably pointing us to the fact that these are also have significant relationships with child maltreatment outcomes. This one is for emotional neglect. You see the same patterns here. So bisexual did reach statistical significance, so increasing the likelihood that these individuals would also experience emotional neglect. But we also see large effect size for homosexual, I don't know, and other identity, but likely just underpowered models there as well. So it's likely if we had a bigger sample, we would find those significant effects. And this is exposure to intimate partner violence. The I don't know category had an increased likelihood of 7.45 times with a very large confidence interval indicating lots of variance in this model, but still statistically significant related to exposure to exposure to verbal intimate partner violence in the home. So many significant relationships were found for sexual identity and a variety of different child maltreatment types. We also wanted to look at the relationships between sexual identity and mental health and substance use outcome in our emerging adults. So this is just a slide here that sort of summarizes all of our significant findings, and I will also demonstrate this using bar graphs as well. But what you can see here is the reference group. So compared to heterosexual sexual identity, you could see for depression, we had if you identified as homosexual, gay, or lesbian, you had 12 times increased likelihood of depression. If you identified as bisexual, 5.3 times increased likelihood of depression, and other identity, 4.32 times increased odds of depression. The I don't know category didn't reach statistical significance, but likely, again, it's a large effect size, so likely an underpowered model. You can see significant findings for bisexual and anxiety. And again, moderate effect sizes for other types of sexual identity, but didn't reach statistical significance. We had significant findings for homosexual and at-risk cannabis use, and bisexual identity and at-risk cannabis use, and also bisexual identity and at-risk alcohol use as well. So these are those findings graphically. So you can see that these are our findings for depression. So we have 4 sexual identity with types with depression. This is for sexual identity and anxiety, sexual identity and at-risk cannabis use. So we have 2 significant models there, and sexual identity and at-risk alcohol use. So from there, we really wanted to understand the moderation effects and whether or not looking at the combination of sexual identity and child maltreatment had a further impact on whether or not mental health would be impacted for these youth and emerging adults. And so we did find in the interaction effects, we did find those trends that the experience of child maltreatment along with sexual identity did make the mental health outcomes worse. So we know that those relationships exist, but having both of those together really did show an elevated outcome. Here, we're just looked at stratified models because, again, as mentioned, we had difficulty due to power. So we have our stratified models here, and we're looking, these are our data just from among those with any child maltreatment. And you can see many, the bolded findings there are indicating much elevated levels for depression, and anxiety, and at-risk cannabis use and substance use from the slides previous, when we're both looking at child maltreatment and sexual identity together, and not just sexual identity or child maltreatment on its own. So our summary is that compared to identifying as a heterosexual, sexual identity of homosexual, gay, or lesbian, bisexual, or other identity were associated with increased likelihood of child maltreatment, but also increased likelihood of poor mental health and substance use, and having these experiences together had the largest impact. Some of our models were more, we found more significant findings for depression and anxiety and fewer for at-risk cannabis use and alcohol use, but we definitely found significant findings for substance use as well. And the trend suggests that the association between sexual identity and poor mental health and substance use outcome are worsened in many cases when child maltreatment is also present. So the strengths of our study is that it's a community sample of emerging adults, including both urban and rural settings. As I mentioned before, often university samples or social media are used to recruit these samples. We're able to find a high-quality community sample. It's Canadian, it's self-reported surveys, it's multi-wave longitudinal data. We included several sexual identity types. We tried to look at as many as we could without having to collapse any of those categories. And so we were able to do that for most cases. And we also looked at several child maltreatment types instead of only looking at sexual abuse, which is often the case when looking at sexual identity. And also in Canadian nationally representative samples often have 3 types of abuse: sexual, physical, and exposure, and don't look at emotional maltreatment or neglect. Limitations, of course, it's just a sample from Manitoba. It's not nationally representative. It's a community sample, so we don't generalize beyond we can't generalize beyond our sample. And also we had the plan of looking at gender identity in addition to sexual identity, and with our sample size, we had inadequate statistical power. Our study implications is that preventing child maltreatment among all children, including children identifying as other than heterosexual, is a public health priority. Promoting and maintaining safe environments for all children is essential for healthy development and to reduce mental health disorders and at-risk substance use. So one of our recommendations is to invest in public education and resources that normalize and promote the acceptance of all sexual identities and remove this intolerance for violence against children and youth. So we can really focus on parenting and understanding sexual identities related to development in adolescence and in emerging adulthood would be very helpful to reduce violence for children. So again, awareness of that relationship is also very important for clinicians, and also using evidence-based methods to recognize and safely respond to child maltreatment in these clinical settings. Policies and resources, parenting programs that educate caregivers about child's development, including sexual identity, may prevent and reduce childhood violence. We also need trauma-informed and violence-informed policies to prevent violence against children and youth who identify as lesbian or gay, bisexual, or other sexual identities, and investing in these public education and resources that normalize and promote the acceptance of all sexual identities are needed. So in terms of future research, we are actually have extended these findings to a nationally representative study that are currently under review. So I wasn't able to present those data to you today, but we were able to replicate that using a larger sample that was nationally representative, looking at a few different outcomes as well. So that will be data that we can present soon. But we also need to further invest in nationally representative data and that can inform policy and practice. We need to make sure that we are putting recommendations forward for these national surveys that look at all types of child maltreatment, and also we can safely look at these types of child maltreatment experiences in younger children. So we could be doing that in nationally representative data. And also collecting longitudinal data is really important. We were fortunate that we were able to have multi-waves of data from 2017 onwards up to 2022. So that was able to include some COVID data in there as well. So having these longitudinal data sets allow us to quickly move when we need to for those reasons. And then also we weren't able to look at gender identity. So we need larger samples to do that, and sometimes even larger samples than what's included in StatsCan. We even find those power issues with StatsCan data sets that we've been using as well. So those are some future directions to help us get even better understanding to fill current knowledge gaps that still exist. Before I stop, I also just wanted to mention that as many other recipients have said their appreciation for to be awarded this award. And so I just wanted to acknowledge that I was able to renovate my lab in part with the funds from this award. So I just wanted to share some photographs for some of the spaces that were renovated. So here we have a common area at the entrance of our lab. We have a small collaboration room for small meetings. Here we have my office, and then 2 rooms here with individual workstations for our students and our staff who work in the lab. And then we have a collaboration room or virtual classroom that we have here with a small conference area, and able to have our virtual classroom and conferencing across other labs. So a lot of the funding from the award that I received, I did use to renovate the space, which is greatly appreciated, because anyone who knows about renovating spaces, there's not a lot of funds that qualify or that are eligible for that. So I wouldn't have been able to do this without this award. So I'm grateful, my students are grateful, my staff are grateful that we can now work in this space ongoing. Thank you.
Thank you so much, Dr. Afifi. Your new space looks lovely, and such an important topic that you're digging into. So thank you for all that you're doing.
Thanks.
So now we will move on to our 2019 recipient, Dr. James MacKillop. He actually, unfortunately, is not able to be with us today. He had a conflict come up, but I did have the opportunity to pre-record him. So I will go ahead and share my screen and play his presentation. So just bear with me for one moment. All right, and we'll get into it.
Hello, everybody. It's great to be with you today. I was greatly honored to be the 2019 recipient of the Royal-Mach-Gaensslen Prize and feel very honored to be in the company of the other award winners over the years. It's my real pleasure to share with you a snapshot of my research program today, and I appreciate the opportunity to do so. What I want to start with in doing so is acknowledging some of the great scientists and trainees that I've worked with over the years, and these individuals are contributors to the science I'll be presenting today. This includes Dr. Larry Sweet at the University of Georgia, Dr. Jim Murphy at the University of Memphis, and Drs. Wei Deng and Dante Duarte at McMaster University. I've also been very privileged to work with really outstanding trainees, and some of the work I'll be presenting was led by postdocs in my lab, specifically Drs. Victor Martinez-Laredo, Dr. Alba Gonzalez-Raz, and Brian Bird, as well as a PhD student in my lab, now Dr. Emily MacKillop, and two really outstanding staff members, Carly McIntyre-Wood and Kyla Belizario. I'm very fortunate to be part of a big team doing a lot of different interesting projects. I want to make sure I express my thanks to everyone who contributes to this work. So at the heart of my research program are four pillars. I trained as a psychologist and as a behavioral economist, and so behavioral science is at the heart of my work. What I mean by behavioral science is understanding the basic behavioral determinants of addictive behavior. This includes different traits, states like craving and negative emotionality. It includes cognitive contributions, and basically is the study of the psychology of addiction. In addition to behavioral science, I use cognitive neuroscience to try to understand the neural underpinnings of the phenomena I study in the behavioral lab. So we use task fMRI, resting state fMRI, and other neurocognitive paradigms to understand what's going on inside the brain when people are making decisions about addiction. At the other end of the spectrum, we translate our findings in the behavioral lab forward into clinical research. This includes studies trying to develop the next generation of diagnostic tests, prognostic indicators, new treatments, and in some cases, applications to public policy. And under each of these three pillars, I'm interested in how genetic variation gives rise to variation in behavioral processes, variation in the brain, and variation in clinical outcomes. And so these are the four key pillars of my research program. And I'll give you an overview before we get into some actual recent results. So in terms of behavioral science, one of the most common methodologies that we use is observational cohort studies. These are studies in which we study groups of people over time. This is just one example of a study of high-risk young adults from my lab. On the bottom x-axis, you can see observations over time from 2018 to 2024. And on the y-axis, you can see all 17 waves of data collection that we've done so far. These waves are what we call squids, as illustrated by the bottom figure that shows the distribution of participants participating over time. And we look at the interrelationships between variables in the brain or in behavior in relation to substance use using cross-lag Pinel models like the example you see on the lower right-hand part of the screen. In terms of our brain imaging research, these are images from some of our recent studies using a wide variety of different neurocognitive and neuroimaging paradigms. We use task fMRI, in which we look at neural activity during different experimental tasks. We also use resting state fMRI to see what the brain is doing at rest in relation to addiction. And we're using both traditional and advanced brain morphometry to understand the structure of the brain in relation to addictive outcomes. When it comes to our genetic research, this work started in my lab all the way back during the candidate gene era, and the top left figure shows you our very first study in which we identified a promising candidate gene in relation to impulsivity. That's changed a lot over the succeeding years, and indeed now about 2 decades. Now we're mostly doing genome-wide association studies with much larger samples, and to do that, we're partnering with companies like 23andMe, in which we're able to give our various behavioral paradigms to large numbers of individuals, as illustrated by the figure on the right side of the screen, which was our delivery of our impulsivity test to more than 25,000 23andMe customers to try to get a sense for the underlying genetic architecture of self-control. Last, in terms of our clinical applications, I'm very privileged to collaborate with a number of different clinical programs. That includes at my home hospital, St. Joseph's Healthcare Hamilton, where we collaborate with Men's Addiction Services Hamilton, our Concurrent Disorders Program, and Womankind Addiction Services. But I also have the opportunity to work with Homewood Health and its clinical programs, including the main hospital in Guelph and its other inpatient facility in Victoria, British Columbia. And then the last piece of the puzzle is we work closely with Correctional Services of Canada to do research on the intersection of criminal behavior and substance use disorders, including collecting data from federal institutions to understand processes that may contribute to this intersection. So at the other end of the spectrum, we're really trying to embed our research in these real-world, complex, high-volume clinical settings. So where does behavioral economics come into this? Well, behavioral economics is the theoretical framework that I apply to a lot of my research. Behavioral economics basically reflects the hybridization or the integration of psychology with economics to understand how people make decisions, what they value, what their preferences are, and what biases can creep into their decision-making that can lead to problematically irrational behavior. This is a particularly good framework for understanding addiction because in many ways, we're studying individuals who are struggling with making persistently bad choices to keep using substances despite negative consequences. And unfortunately, being unable to consistently make the choices that lead to healthier outcomes. So focusing on decision-making, choice behavior, and the dysregulation of choice behavior is a really compatible framework for understanding addiction. In particular, we focus on three different domains or factors when it comes to behavioral economics and addiction. We focus on an individual's tendency to overvalue smaller immediate rewards relative to larger delayed rewards, a process called delayed discounting. We study excess valuation of addictive drugs, a process called drug demand. And we also study the absence of alternative reinforcers as the third factor in terms of determining substance use. And these are the three principal foci of my research program. I'm just going to focus on one today to sort of highlight the kinds of work that we're doing in my lab. And that's going to be the overvaluation of drug reward. So let's start with the behavioral science of drug reinforcing value. And before we get started, I just want to provide a little bit of a primer in terms of how we measure the value of a drug in our behavioral science studies. For any of you who have taken microeconomics as an undergraduate or have done any kind of economic coursework, this probably looks pretty familiar, but I know a lot of people haven't done that, so it may be unfamiliar. But this is fundamentally a demand curve. It's the representation of the consumption of a commodity relative to its price. It could be standard drinks of alcohol, it could be grams of cannabis, it could be hamburgers, it could be crayons. But fundamentally, what we're looking at is the relationship between the commodity in terms of its consumption on the y-axis, going down as a result of the increase in price on the x-axis. And you can see two prototypes of people here, one exhibiting higher demand for a commodity and one exhibiting lower demand for a commodity. And the value of these demand curves is that they provide us with multiple indicators of how much a person values a commodity. This starts at the beginning when prices are low, at the beginning of the demand curve. We call that intensity when there's zero price or very low price. So that's maximum consumption. And at the end of the demand curve, we have breakpoint, which is where a person will no longer consume the commodity because the price is too high. In between the two, we have an indicator called Pmax, which is where the demand curve transitions from being generally insensitive to prices to sensitive to prices. And the last thing that we can do is we can actually model the curve of the demand curve as a way of measuring how much a person values something. Again, it could be alcohol, it could be cannabis, it could be cocaine, but this allows us to very precisely define exactly how much a person values consumption by juxtaposing how much they want to consume versus how much it costs. Now, when we've applied this to studying substance use, what we found early on is that actually this approach really maps onto substance use behavior quite well. These are data from one of our early studies, dividing individuals into whether or not they were hazardous drinkers, defined as having an audit score of 8 or greater, or non-hazardous drinkers, defined as having an audit score lower than 8. And in this sample of overall regular drinkers, you can see that the hazardous drinkers show much higher alcohol demand, starting at higher levels in terms of intensity, and also being flatter over time, and generally being larger in terms of overall consumption across different prices. And so just to give you a sense of what this looks like across the literature, since we developed the measure, this has been applied to a variety of different drugs. These are all findings from my lab looking at alcohol demand or tobacco demand, cannabis demand, and cocaine demand. And what we see over and over again is there's this prototypic relationship in which at zero or very low prices, people will consume quite high levels or report wanting to consume high levels of their preferred substance. But as the cost goes up, consumption goes down, and we're able to model those cost-benefit relationships quite precisely. What we've most recently done is start to aggregate the findings across the literature. As more and more groups are using these tests and investigating the relationship between demand and substance use, we wanted to see how robust these relationships were. So we did a number of meta-analyses looking at the most common drugs that have been studied so far. There's a lot of data coming up on the next slide that reports what we found in these meta-analyses. But to give you a sense for what I'm going to show in a second, this is a synthesis of what we found in terms of alcohol, cannabis, and tobacco. And you can see that across studies, we were able to aggregate lots of participants with sample sizes as high as nearly 14,000 in terms of the alcohol studies. And in the next figure, the denser or the darker the figure that you see, the larger the sample size. And in terms of the statistical significance, you'll be able to tell whether an effect across studies was significant based on the presence of either a circle or a circle with a cross through it, indicating that it was highly statistically significant. So synthesizing across studies in 3 different papers, what we did was look at to what extent were there significant and large magnitude associations between the value of a drug and a person's involvement with the drug. You can see we subdivided into the quantity of consumption versus problems resulting from the drug, or how dependent a person was from the drug. And there's a lot going on here, but one of the clear take-home messages is that across studies with lots of participants, for a number of these indices, which are indicated on the lower x-axis, we see large effect size relationships, sometimes very large effect size relationships exceeding more than a standardized effect size of 1.0. In most cases, we also see that these are statistically significant. The one exception seems to be that Pmax really does not appear to be nearly as relevant as some of the other indices. When it comes to the value of addictive drugs, intensity of demand, Omax, or how much a person is willing to spend on the drug, and alpha, that elasticity value that measures a person's cost-benefit sensitivity, are the 3 biggest indicators that are associated with an individual's level of substance involvement. So we see that this process of making cost-benefit decisions about a drug is related to the level of involvement. But the next question for us was, what's happening in the brain when people are making these decisions? What are the neural underpinnings of making choices between using a drug and not using a drug based on escalating levels of cost? And we started doing this work some time ago. This is the original paper that we published developing basically an fMRI paradigm of our alcohol demand methodology. And to do this, what we basically did was conceptualize the kinds of choices that are being made as being in 3 different categories. And we used the traffic lights convention to think about these conventions. The green light basically was the go category, in which people were completely insensitive to the costs at very low costs and were consuming the highest amount. The amber light was our elastic demand category, in which people were sensitive to the costs, but they were still consuming alcohol. And the red light was basically the stop category, or the suppressed category, in which people were no longer willing to consume the drug, in this case alcohol, because the costs were too high. And in this first study, we found that there were some clear orderly differences in terms of brain activation based on these different kinds of choice preferences. And in particular, subunits of the prefrontal cortex, especially the medial PFC and the dorsolateral PFC, were implicated in alcohol demand decision-making. And then the other region that seemed to be really critical was the anterior insula, which is responsible for a lot of interoception and has also been implicated in drug craving and other forms of motivated behavior. So we started to see a clear ensemble of regions that were implicated. But what we didn't know in this first study was, how does the brain of an individual who's addicted to alcohol differ in terms of these choices during these kinds of tasks? So the most recent study that we've just finished has asked exactly that question. We recruited a sample of individuals with alcohol use disorder from the community. We recruited a demographically matched group of recreational drinkers. And then we undertook a case control study comparing cases, the individuals with AUD, to controls, our demographically matched control group, and everyone underwent an fMRI scan using our paradigm. These are the data behaviorally from the fMRI scan. You can see the darker colors reflect the AUD positive group and are substantially elevated in terms of the overall demand curve. But you can see also those individual indices, intensity, elasticity, Omax, and breakpoint, are all elevated significantly also. So we see behaviorally in the magnet, individuals with an alcohol use disorder show significantly greater value of alcohol, just like the behavioral studies. And then in the brain, what we found was that it wasn't a single location or a small number of locations that differentiated. Actually, it was a whole ensemble of regions that were implicated and also differentiated. And I'm not going to go through each of these. I'm going to really zero in on where the biggest effects were. So when people were choosing to drink the most during that inelastic portion of the demand curve, we saw significantly greater activation in the right lingual gyrus in the AUD positive group. And interestingly, we saw the opposite. We saw significantly lower activation in the left lingual gyrus in the AUD positive group, as well as lower activation in the inferior frontal gyrus, which is implicated in behavioral inhibition. In terms of other regions, we found bilateral activation of the anterior insula, significantly elevated in the AUD positive group compared to the controls when people were making those price sensitive or elastic choices. And the same was true when we used a different indicator, Omax, where again we saw significantly greater recruitment of the anterior insula in the AUD positive group compared to the control group. What was really interesting was that when we looked at these regions in particular, and two especially, the right lingual gyrus and the right anterior insula, these regions actually differentiated our alcohol use disorder positive group from our control group very precisely, and indeed exceeded the standard benchmarks for a diagnostic test. So it seemed like these regions in particular were highly sensitive to group differences in terms of how the brains of individuals with alcohol use disorder differ from individuals who are merely recreational drinkers. What about genetics? We know that about half of the variation in risk for developing substance use disorders is attributable to genetics. And as more and more studies have enrolled larger and larger samples and scoured the genome for variants that are implicated, more and more are being identified as being statistically significant, but we rarely know how those variants are functionally implicated or what the mechanisms of those variants are. So we've tried to use behavioral economics to better understand how genetic contributions contribute to differential risk. And just to give you one example, these are findings from a recent study in which we looked at alcohol polygenic risk scores and then tried to use our alcohol valuation measure to understand how these PRSs were implicated in substance use risk. So just to give you some background, polygenic risk scores are basically aggregations of the individual polymorphisms or variants that are implicated with the condition, and they're then integrated together into a measure of risk. And so we looked at two very established PRSs, one for drinks per week and one for the audit consumption score. And then in one of our cohort studies, in which we also had DNA, we conducted what's called a PheWAS study, or a phenome-wide association study, in which we looked at these polygenic risk scores across a wide array of phenotypes. And you can see those color-coded at the bottom of the screen. We looked at personality, we looked at impulsivity, we looked at psychiatric comorbidities, we looked at reasons for drinking. Our focus here was really on drinking, obviously. And we also looked at alcohol reinforcing value. And what you can see in these two figures is on the x-axis, the different phenotypes that we looked at, and on the y-axis, the negative log 10 p-values that indicate how statistically significantly a phenotype was implicated. And what we saw was that, just as we expected, the drinking variables were indeed linked to the alcohol-related polygenic risk scores. So there was internal validation there. But the next variables that were most predictive of those of the polygenic risk scores were our alcohol reinforcing value measures: Omax, breakpoint, elasticity. Those really showed the most robust relationships And that was very different from many of the other variables, such as self-reported impulsivity traits or personality traits in general, and even things that are quite commonly implicated, like comorbid psychiatric symptoms. So what we see in this study is that by understanding how much a person values alcohol, we can see that one of the ways that the polygenic risk scores are linked to alcohol involvement is via this biobehavioral process of overvaluation. The last piece of my work that I'll say about today is how we then bring these paradigms and perspectives to bear on the clinical research enterprise. So in this case, what we're really trying to do is focus less on disentangling what's happening in the brain when people are making these choices, or how these kinds of preferences can inform or illuminate genetic contributions to addiction. We're interested in whether or not these indicators can illuminate clinically relevant behavior, either in community samples or in clinical samples. So in one example, we used the indicator of alcohol valuation again to try to understand alcohol use in a cohort of emerging adults. And as many people know, heavy drinking is a major public health issue in emerging adults. It's associated with physical assault, sexual assault, academic and vocational adverse outcomes. And although many people do ultimately age out of this behavior, it's not clear what are the best predictors of these either salutary changes over time or persistent patterns of heavy drinking in young adults. So in one of our cohort studies, we examined a group of a bit over 600 individuals over 6 waves of data collection, and we were interested in looking at what variables predicted who changed their behavior over time. This figure here shows what we expected to happen, which was that from the start of the study, when people were in their early 20s, we expected the number of heavy drinking days per week to go down successively over time. And as you can see, that happens wave after wave until people have gone down on average by about a third about 3 years later. When we looked then at our alcohol demand indicator, our behavioral economic indicator of alcohol reinforcing value, we used those cross-lag panel models that I described earlier to model the overall association, which is what you see highlighted at the far left, and to then look at the cross-lag patterns to see which variables predicted each other over time. And what was critical, we're going to focus here on our OMAX indicator, or the maximum amount of money a person would be willing to spend for alcohol. What we found was that wave after wave, that variable predicted the drinking at the next wave, but the opposite was not true. Drinking did not predict the OMAX at the next wave. So rather than merely being a correlate of heavy drinking, what we found was that this measure of alcohol reinforcing value predicted subsequent drinking at each subsequent wave. As one last example of our clinical research, we're also trying to integrate these indicators into clinical research focusing on novel treatments for addiction. And one of the treatments that we feel has high promise is deep repetitive transcranial magnetic stimulation. And we are currently completing a pilot trial of deep RTMS for cannabis use disorder. For those of you who are not familiar with this intervention, it is the use of powerful magnets to stimulate regions of the brain, specifically individual nodes, but also networks within the brain that are implicated in a given psychiatric condition. And deep RTMS, as opposed to conventional RTMS, uses specially configured helmets that have magnetic coils that can stimulate the brain at deeper areas and using a more network-focused approach. And this approach has been Health Canada approved and FDA approved for tobacco use disorder, as well as several other conditions. And we believe it has considerable promise for cannabis use disorder. At the bottom of this slide, what you can see are our preliminary results to date in terms of changes in cannabis use. You can see prior to the onset of the intervention, individuals were consuming about 2 grams of cannabis a day, and that went down to close to 0 by the end of the treatment period. That was actually a 90% reduction from baseline to the closing of the intervention period. So overall, we're seeing a very good response to this intervention. And in terms of understanding what mechanisms are responsible for this, if we use our behavioral economic reinforcing value task, we can see that there are commensurate changes in how much the individuals value cannabis as a commodity. In dark green, you can see their cannabis demand curves at the enrollment to the study, which look just like those other demand curves I showed you before, high to start with, but going down in relation to prices going up. And then on the bottom, in the light green, you can see the cannabis reinforcing value at the end of treatment, and you can see that that's dramatically lower and pretty quickly gets to 0. Indeed, we saw a greater than 80% reduction on each of our cannabis reinforcing value indices, suggesting that a substantive part of the positive effects of the deep TMS are because of the effects on cannabis reinforcing value. Obviously, this remains to be verified in larger subsequent follow-up studies, but this illustrates in both observational populations and clinical populations how these behavioral economic tasks can provide clinically useful information and illuminate some of the mechanisms underlying substance use behavior change. So just to offer some final conclusions, I hope that after this presentation, you have a sense for the value of a behavioral economic and neuroeconomic perspective on understanding substance use disorders. This framework provides a powerful way of precisely measuring the value of a drug, the value of immediate reward, the value of alternative rewards, and these processes play key roles in the development of substance use disorders, as well as their maintenance and ultimately their successful treatment. I want to finally wrap up by thanking the funders of my work, who've all provided unrestricted research funding to support this research, and to thank once again the Royal-Mach-Gaensslen Committee for selecting me in 2019, and more generally for creating the Royal-Mach-Gaensslen Prize for Mental Health, which I think is a really critical contribution to supporting mid-career scientists in Canada. Thank you very much.
All right, so thank you to Dr. MacKillop, who cannot be here, but that was a fascinating discussion or presentation. So now we are about 2 hours in, so we're going to give everybody a chance to have a quick bio break, grab some water, coffee, whatever you desire. So I will just start I have a timer that I'll put on the screen so everyone knows when we are all returning. So I will do that now, but we will reconvene at about 3:07. Thanks, everyone.